Folks, a fine day to each of you. As I say, I hope your day is as good as possible. And since you've found yourself here, I think it's a really good day because right in front of you is your extended family. And every 2 weeks, we get to smile at each other, cry with each other, tell of our victories and tell of our sadness. It's super important. So welcome to another Expert Talk series. We're so proud that you know that we're here for you. Thank you. And with that, Brian?
Perfect. Now I'm going to introduce the speaker. The speaker today is Dr. Sandra Banack. Dr. Sandra Banack is the senior scientist at the nonprofit Brain Chemistry Labs in Jackson Hole, Wyoming. She received her Ph.D. in Integrative Biology from the University of California, Berkeley, working in the Museum of Vertebrate Zoology. Dr. Banack left a full professor position at California State University, Fullerton, Department of Biological Science, to join the nonprofit Brain Chemistry Labs, where their only goal is to change neurodegenerative disease patient outcomes. She is among 4 scientific researchers at the center of global consortium of 50+ scientists focusing on determining causes and developing treatments for ALS, Alzheimer's, Parkinson's, and other brain tangle diseases. Thank you once again, Dr. Sandra Banack, for taking time and effort, and thank you very much.
It's nice to be here with you all. I also note that Dr. Cox was able to join us, actually, and maybe would like to say a couple things. Dr. Cox?
Well, just it's a real honor to be with all of you. Please know that there's a group of cowboys and cowgirls up in Wyoming thinking about this every day, and we are totally determined to get ALS out of the map of the world. So we're so grateful to you. And I just thought Dr. Banack gave such a brilliant talk in Switzerland on Friday at the International ALS MND conference. I thought it'd be fun for you to all hear from her. So let me turn time back to her. So many thanks, Sandra.
Well, thanks. We just came back from this Congress for MND ALS, where there were 1,300 scientists and clinicians and a few ALS patients who were there. And we were able to share our research and discuss what was going on. So what I thought I would do is go ahead and talk to you about the presentation that I gave and show you that and walk you through it of the latest research in diagnosing ALS. So if I can get my screen up here going, I will share this and turn it into a slideshow. Can you see that all right?
Yes, we can.
Okay. So what we've developed here at Brain Chemistry Labs is a robust microRNA ALS blood diagnostic test. And I'm going to walk you through the details of this, and then at the end, take any questions that you might have. I'd like to acknowledge all of the contributors who have contributed to this work that I'm going to present. Some of us are from Brain Chemistry Labs, but Dr. Elijah Stommel is a neurologist at Dartmouth. Dr. Paul Maida and Han Moon are epidemiologists and doctors at the Center for Disease Control, the CDC. Dr. Hiroshi Mitsumoto is a doctor at Columbia, a neurologist, and Dr. Paul Cox, who's here with us today, had much of the vision for what we're doing. I'm grateful to him and for all the support. So I don't have to tell this group, but diagnosis takes too long. And unfortunately, a very large proportion of patients are misdiagnosed along the way. Somewhere between 3 and 68% get a misdiagnosis along this path. And the cost of diagnosis is also not irrelevant. There is a profound cost, and more if there is a misdiagnosis along the way. And for all of these reasons, we sought to make a improve the process and to create a diagnostic that would help ALS patients. Our only goal at Brain Chemistry Labs is to change patient outcomes. And so in this search for a biomarker, a diagnostic biomarker, as you're aware, ALS shares these early stage symptoms with a lot of other neurological diseases. And some of these diseases are called specifically ALS mimics, but there are other things, and anybody who's been diagnosed knows that there is overlap between many of these neurological diseases. The goal would be to diagnose ALS very early and therefore be able to intervene early before neurons are lost and before much of the damage is done. There is a greater chance of success for both the diagnostic, for both the prognosis problem, but also in being able to potentially reverse it, stop it, and to create new drug therapies. We all, I think, would agree that a blood-based biomarker could very much aid neurologists in diagnosis. And so in our search for that, we wanted it to be blood-based, not a cerebrospinal draw, which is, of course, a lot more traumatic. We wanted a biomarker or a diagnostic test to be unique for ALS and not to have overlap with other potential mimics. And something that had to be completely robust and repeatable, something you can do easily, get a blood draw, and be able to aid neurologists in diagnosis. And so we began focusing on a group of particles within the blood called extracellular vesicles. These are abundant in every body fluid that we have. You can find them in saliva, you can find them in blood plasma, you can find them in urine. And so because they're abundant, they seem like a good target. We know that these extracellular vesicles, or EVs, contain proteins on the surface of them that reflect the tissue of origin. And what we wanted to do was to create a diagnostic test that had these extracellular vesicles that came from neurological tissue rather than from your kidney or your liver. And so I'll show you how we did that in our approach to that. We know that within these extracellular vesicles is a cargo that is non-random, or in other words, it's diseased influenced. So these extracellular vesicles are packaging into them little pieces, and they're thought to be communication vectors between cells and to pass information on between cells. And therefore, what's put inside of them is unique. We also know that there are encapsulated in what's called the lipid bilayer, which protects them and protects that cargo so that it is not degraded by enzymatic or other things within the body. And so these extracellular vesicles, and this is somewhat technical, but there's really sort of three classes. And of these three classes, we were focusing on the small end, these things here called exosomes, which are about 40 to 150 nanometers in size. And these can be characterized in a variety of ways, which we used, again, focusing on these very small ones. And this is a nano-tracking analyzer that we use to be able to quantify what are the particle size and how many do we have of these different particles. And so again, we're focusing primarily on these exosomes, but the other extracellular vesicles are also contained in our samples. Within the extracellular vesicles, as I mentioned, they're packed with things called proteins, packed with RNA or DNA, which are called nucleic acids, or they can have in them lipids. And what we wanted to focus on, the cargo of interest to us, is something called a microRNA. And these are very tiny pieces of nucleic acids. They're about 18 to 22 nucleotides long, so that's very, very small. These are little pieces of RNA that are non-coding, so they're not used to be turned into proteins, but instead they suppress the translation of the RNA into proteins. So they stop protein production, in other words. There is evidence, as I mentioned, that they're non-random loading, and therefore they can be disease specific and they can reflect disease processes. There's also some evidence that they can influence disease progression. These little tiny particles get put out of the cell and they're transported to the next cell, and they can have information that can increase disease progression by passing along information that is detrimental, but they also can pass along positive information. And so for these reasons, we went into a process of discovery, and the first aspect of that discovery is called EV isolation. And so our goal was to take blood plasma and isolate these EVs through a PEG precipitation process, and then enrich them based upon the neural proteins that are identified on the outside of these EVs. And then we did a lot of work to characterize them and made sure that we knew exactly what we're looking at and the size and the characteristics of these EVs. The next step that we went through is a process called next generation sequencing. And the idea here was to identify everything that is RNA, and particularly the small RNAs is what we were interested in, that are present as cargo inside these EVs. And so after collecting the EVs, we did an RNA extraction. We actually sent these to QIAGEN to do a library preparation and what's called this next generation sequencing. And from these, we were able to identify 1,700 small RNA species. So they contain a lot. Within that were 419 of the specific ones that we were looking at called microRNA. 101 of these showed differences between ALS patient blood plasma and control blood plasma. And we selected 35 of those that had the greatest differences between controls or healthy patients and ALS patients for additional analysis called qPCR. Now, when we think of qPCR, basically think of COVID. When you were getting a COVID test and they swab your nose and then they test and say, do you have COVID or not? They were doing primarily qPCR. This is just a very specialized and very sensitive way to see what's inside and to see these small particles of DNA and or RNA is what we were looking at. And I show you this to say that once we were able to identify some microRNA that were of interest, 8 of these we found to be very diagnostic of ALS. And the question I always get asked among scientists is, are these microRNA that you see reflective of ALS or reflective of neurological processes? And what you see on the right are basically the answer to that is yes. There's a lot of processes that we know are part of ALS or part of neurodegeneration in general or specific to ALS that these microRNA are part of. Now, a microRNA can affect many different proteins and many different processes, but at least there's a good correlation here. So what we did is we tried to measure what's called fold regulation, which is how much of a difference is there between an ALS patient and a healthy blood plasma sample. And to do this, we were able to collect some blood plasma samples from a clinical trial. And anybody or any of you who have ever donated blood plasma or been involved in a clinical trial, I have to say thank you. From the bottom of my heart, that really helps us to get information to understand the disease process and how we can help. And so this particular clinical trial was being run by Elijah Stommel, and Brain Chemistry Labs had a very large part in this. And the samples, the blood plasma samples that came in from this trial, we were able to take and to look for differences in these microRNA through this isolation process between ALS patients and control patients. And again, we looked at 8 different, what we call microRNA, and we looked at whether they were turned on in the case of these ones or turned off, which is called downregulation or upregulation of these various small particles. And we were able to test 10 ALS patients and 10 controls. Now, these particular samples from a clinical trial were very carefully processed. There was only a single neurologist that identified the diagnosis of ALS in all patients. These samples were processed in a very specific way. Within an hour of collection, they were spun down, they were processed, and they were frozen at -80 degrees Celsius. And so they were very carefully controlled. And so the first thing we did is we looked at, are these microRNA, are they upregulated or are they downregulated? Are they turned on or are they turned off? After we got these results, we then did a second set of 10 ALS patients and 10 controls. Now, these were completely unique and completely different from the other ones, but you'll notice that the numbers that you see, the difference between an ALS patient and a healthy person, were very similar in each of these samples. And this was published back in 2020 in a very prestigious journal called the Royal Society Open Biology. And from that, we decided we needed a bigger sample size. 20 ALS patients, 20 controls is just not nearly enough because, as you know, ALS is a very heterogeneous disease. There's a lot of variability between patients and where it starts and how it moves and how fast it progresses. And so we replicated this again, getting some samples from the CDC and looking at a very much larger sample size. And so you'll see the very first experiment we did here, the second experiment. Here's the third experiment, now with 50 ALS patients, 50 controls. These samples from the CDC represented identification of ALS from all over the country, different neurologists identifying the disease. It reflects, in most cases, a phlebotomist came to the home of the ALS patients, withdrew blood. Those were then shipped out after a period of time to a center by Thermo Fisher, who then spun them down and froze them. But because they were all treated differently, they had different time periods, they were from all across the country, we were actually surprised that you can see the exact same pattern. Everyone that was upregulated the first time was upregulated the second time. The ones that were downregulated were also, and all within a very similar range. This gave us great hope that we had a robust and repeatable sample. And so we decided to replicate this again. In science, you have to be able to repeat things, particularly if it's going to be a test for ALS patients. So we began undergoing a fourth replication. This time we had 150 samples from the CDC and some additional samples from the clinical trial that we additionally looked at and control samples. And again, what you see with this large sample size of 264, all of these microRNA were again within the same range that we saw prior, upregulated or downregulated in the same way. And that tells us that we have something that is repeatable, that it's robust, that we can count on this, and that these microRNA are very reflective of the disease itself. And it's upstream from what's mostly looked at during the diagnosis of the disease. So now we had a total of 189 ALS patients, 220 that were non-disease controls, for a total of 409. But in this fourth replication, we were able to add something very important, and that is disease mimics. So from Dr. Hiroshi Mitsumoto, we were able to find primary lateral sclerosis patients, very similar onset of early symptoms, but a difference in the progression of the disease and how it moves forward. We were also able to take some Parkinson's patients, which are considered an ALS mimic, particularly in these early stages. So now we have 471 total samples, and I'm going to show you what the primary lateral sclerosis and the Parkinson's showed us in just a minute. So what you're looking at is one microRNA here called 10B5P. And you can see the controls here and the ALS patients here. You can see a very real difference both in this was the mean and the variability that you see within samples. The controls have a lot more variability than the ALS. And you can compare those to both the PD and the PLS just to show you how much variability there are. This particular 10B5P was the microRNA that contributed most to the next things I'm going to show you. The second one that contributed the most was a microRNA called 4454. And again, you can see very large differences here between what we're looking at of these different, both the neurological controls and the healthy controls. And we were able to take these 8 microRNA that we're looking at and decide which ones contributed the most, because maybe we only want to look at 1 or 2. And what we did, we looked at all 8 because we thought that would be the best to accurately diagnose ALS. And so the real question is, can we differentiate between ALS and ALS mimics? And the answer is definitively yes. What you're looking at in here are ALS patients versus healthy controls. And when you look at the microRNA, every one of them has a very large difference between here that we can look at and identify statistically. When we looked at the Parkinson's versus ALS patients, only 5 out of the 8 had a very large difference or were significantly different in what's called a Mann-Whitney statistical test. And a couple of them were not, but it was interesting to see that you could get different in ALS patients versus Parkinson's patients. The pattern of regulation, whether they were downregulated or upregulated, were different. And that was important. The next thing we looked at were the primary lateral sclerosis patients versus ALS. And every one showed a very large difference between those 2 groups. And you'll notice here, all but 1 were downregulated in primary lateral sclerosis. And so again, what this tells you is that you can very definitively tell the difference between an ALS patient and a primary lateral sclerosis patient or a Parkinson's patient or somebody who does not have these neurological symptoms. And these samples, again, came from our colleague, Dr. Hiroshi Mitsumoto. Without collaboration among scientists, this work could never be done. And so the next part of this process of discovery and identifying a diagnostic test is to understand things like sensitivity. And what sensitivity is on a test is saying if somebody has the condition or has ALS, can the test identify the difference between those who have does the test positively indicate for those that have ALS and not for those who don't? Specificity is the opposite. If you don't have the condition, can it give you a true negative? But then there's pieces also called positive predictive value, which says that if we get a positive test, does it adequately differentiate between somebody who has the condition and somebody who doesn't? And negative predictive value says if we get a negative test, does that adequately reflect a true negative condition? And so this is a little bit technical, but basically this is a test that we use to look at sensitivity and specificity. And for a scientist, the details here are very, very important. We used the training data from the third replication that we did, and then this 4th replication was used to test that and see whether we could come up with an algorithm that would differentiate reliably between these. We found that we had a sensitivity of 97%. Our specificity was 95%. And our predictive values were 94 and 98%. That's reasonable. When we compare what we found with other possible biomarkers that are being presented as biomarkers for ALS, The big one right now is known as neurofilament light. It is used now in clinical trials to show whether or not the disease is progressing rapidly, or whether or not the drug that's being tested can actually halt the disease process. And if you look at this neurofilament light, that our sensitivity and specificity is certainly good, reasonable. Specificity a little higher than anything that's available in neurofilament light. And then we wanted to again look at blood, because anybody that's going in for a test, a blood draw is not quite as invasive as a cerebral spinal fluid draw. And so if you look at this, we're actually getting better results from a blood plasma than can be found within cerebral spinal fluid. And so that was a real positive for us. We then did a second orthogonal method to look at these exact same parameters. And to do this, we combined our data sets for 464 total observations. And what you're looking at in this graph is this line that goes down the middle is basically a 50% line. You could flip a coin and maybe guess whether somebody had ALS or not. If this is the line that you're following, then you have a very bad test. The red line here shows our test, which actually has 97% accuracy, which is considered very good. Using this different orthogonal method, which is called a logistic regression, we were able to get a sensitivity of 95%, 98%, positive predictive value of 98 and 96 for a negative predictive value. These numbers are a little different than the other one because it's a different test, but all within the same range, suggesting that this test is very robust and very sensitive to identifying ALS patients versus controls. If we look at this, what's called the area under the curve, which is 97% effective, and you compare it to blood tests that are in the literature and other possible biomarkers that are out there, again, this falls within a range that is better than what we would normally expect. And when you compare it to what's in the CSF, it's even more pronounced and positive. So again, we think this test might be very positive. So when we look for a diagnostic test, we were able to find a blood-based test. We were able to find a signature of these microRNA, or what we call a fingerprint, that was very unique for ALS. We found that it was robust and repeatable across samples that were collected in many different ways from many different centers and many different neurologists. And so in conclusion, before I take any questions or answer questions, or Dr. Cox can answer questions too, but we've developed a method for a reproducible, robust blood plasma diagnostic test that was consistent with 4 unique patient cohorts, no overlap in samples, 471 total samples. We found that it was highly sensitive, it was specific, and it was accurate. We were able to differentiate between ALS patients and what we call ALS mimics, like Parkinson's disease and primary lateral sclerosis. We are now working towards making this test available to clinics and patients. We think that this is robust enough, it's repeatable enough that this could be a test that, instead of having to wait that 12 months, as the neurologist is considering the symptoms, the progression, and these clinical aspects, that they can just order a simple blood test and get some results back that will help them in giving patients a diagnostic accuracy. And hopefully patients don't have ALS, and there may be other ways that the symptoms that they have can be examined and looked at. As a lab, what we need to move forward are more ALS mimic disease samples. And at the ALS Congress that we were just at, we found some collaborations, which we're very hopeful for. And we're just talking to them now about getting some additional samples. We are working towards making this a very high throughput method. We're a couple of scientists in a lab right here in Wyoming. And yet we're able, and so what we were doing was very technical and very specific, and now we're finding ways to push this through so a laboratory could actually do this to many different patients at a time. And we had a lot of collaborators who, without which this work could never have been done. And so with that, I'm going to give back my screen and see what kind of questions you guys might have.
All right, perfect. Thank you, Dr. Sandra Banack, for your phenomenal presentation. We would now transition to the Q&A portion of this meeting. So my name is Brian Trong. I'm one of the fellows at ALS. I would like to start with this first question from Mindy. First question is, can these biomarkers be used to predict or diagnose disease onset in the pre-manifest genetic carriers of ALS? Furthermore, neurofilament doesn't really cut it for genetic carriers in the prodromal stage of disease.
No, neurofilament light is a measure of neurological distress or neurological damage. As neurons die, they dump out this what's called neurofilament light. It is not the best test for diagnosis. It actually works pretty well for progression and in clinical trials to be able to see whether you're making an impact on the disease process and stopping neurological damage. So that's the primary way that it's used. But it is not the best test, and we're hoping, but it could be used in conjunction with this. What they find on the neurofilament light is that if they take a blood test early on, that you have a high level of neurofilament light, which is higher than other neurological diseases and much higher than somebody that's healthy. Most of the patients in our trial, in the tests that we've been doing, are sporadic ALS. They're not the genetic ones, although there were a few genetic ones in the mix. We think that this is a test that could be used for everybody because it's measuring these specific biological processes that are apparent in the disease as it progresses. And what we'd like to do is to be able to find a way that we could test people not only very early on, but prior to having symptoms. If you have a genetic familial carry a genetic mutation in your family, that you could see whether or not this is something that you might want to be worried about, or maybe it could relieve that anxiety and say you don't have any evidence of this neurological damage. And so that would be basically the best. And I think, you know, Dr. Cox and I have talked a lot, and his dream is to see that maybe when you're 50, you go in for a regular physical and they do a whole panel of neurological tests. So we've been able to nail down this ALS diagnostic test, but now we're working for a very specific Parkinson's test and a primary lateral sclerosis test and an Alzheimer's test and a mild cognitive impairment test, so that you could just go in healthy and take this neurological panel and give you some information of what things might be coming in the future and allow you to make changes before you have any symptoms.
Perfect. Next question we have is from David. Next question is, will this lead to improvements in ALS patients?
Absolutely, in so many ways. I was been talking to an ALS patient over the last couple days, and to doctors who are concerned about patients and the anxiety that is felt. And as you know, if you were waiting for an ALS diagnosis, the anxiety is incredible. And this would essentially give you a negative answer or potentially a positive answer much earlier, when you still have much less neuronal damage. And therefore your quality of life is much higher, and you have time to think and to plan when your body is still healthy, and to be able to make interventions, whether they're drug related or changes in lifestyle at a very early stage. So it absolutely should help patients. And again, that's our goal at Brain Chemistry Labs, is just to change patient outcomes. We want things to be better for you.
Perfect. Thank you. Next question is from Diane. Next question is, how do you get an order for these tests?
That is a great question, and you can't order the test at this moment. What we're doing is we're working with a laboratory diagnostic company who can take the test that we've developed in our small lab and go to what's called a CLIA lab. In order for the FDA to approve it, it has to be, there's all these conditions that have to happen in the lab, and our lab is not CLIA certified, so we can't run tests for patients. And besides that, what we would do is work through a neurologist, where the neurologist or even a general practitioner could say, gee, I think there's a problem here, a neurological issue. We'll order this test, and it would give them information to help with the clinical aspects of the diagnosis to contribute to an earlier diagnosis. But you cannot order it at this time. I think there's a possibility that we can have this out and available to patients within a year. Dr. Cox, do you have anything that you want to add to that?
No, we're moving as fast as we can because we think it will help not only just patients, but also patient families, and then really cut down the number of misdiagnoses.
Thank you.
Perfect. Thank you, Dr. Cox and Dr. Banack. Next question is Carrie and Lynn have very similar questions, so I'll be asking two at the same time. Question is, have you discussed this with the FDA? And what is their input on this as well? If they have any input at all about your studies?
That is a good question as well. And the FDA has been really, really good to us, and in many different ways. And so the first thing the FDA needs is a large enough sample size to move this forward. They have to show that this is consistent in a very large sample size. And typically that number, as we've been talking to laboratory diagnostic tests that would be companies who would be that transition between what we're doing and actually being able to take the blood sample and run them on a large scale, is a sample size of about 1,000. And so I just got notification today that the CDC has agreed to send us absolutely another 300 samples. The notification came today in an email as I've been talking with them about getting additional samples. And we also received in the lab today another 147 controls. So we are moving this forward to hit that 1,000 number. We should be able to do another 600. We're hoping in the very near future, and that will get us over that. And working with a diagnostic company that can repeat this in their own lab in what's called these CLIA certified conditions, they will then petition the FDA for that. And Dr. Cox, feel free to chime in if you have additional things you'd like to add.
Perfect. Next question is again from Mindy. Question is, have you recruited pre-symptomatic genetic carriers of ALS in your test? And for a follow-up, are you planning on recruiting pre-symptomatic genetic carriers of ALS?
So we do not collect the blood samples ourselves. The blood samples are actually collected by our neurologist colleagues through clinical trials and through other measures. At the ALS Congress that we just met, I met with 2 groups that have these prodromal samples of carriers, actually 3 groups, who have blood samples and who have talked to us about sharing those samples to see whether we could identify a possible ALS condition much prior to actually coming down with the disease. So that's in process, but we're not quite there yet. But we are looking at getting those samples. We have to collaborate with others because we can't accept samples directly into our lab in that fashion.
This next question feels like a little similar question, but I'll ask anyways. This is from Jack. Jack is a participant who's diagnosed with ALS, but now has been reclassified as PLS. Sorry about that. And Jack is interested in joining the blood test. Is there a way for Jack to sign up, if possible?
Dr. Cox, do you want to answer that one? He's silent. I'm going to go ahead. Maybe he stepped away, but the answer is that we can accept a blood sample if it comes through another physician. So if you went to your physician and you showed them our published papers, our data, we would be happy to communicate directly with any doctor that you might have, and they could send us a sample. And that sample of PLS would add to the ones that we have already run and give us a greater idea of whether we can differentiate between these. So any sample we get, particularly from those mimics, are really critical at this stage of developing this test, and we would welcome them, but we'd have to work through your doctor. Again, Dr. Cox, do you have anything else you'd like to add? Are you still there? Okay, go on.
Perfect. Next question we have is from Richard. Question is, what ALS disease mechanisms does your miRNA test relate to?
So it looks at oxidative stress. Do you want me to go back to that slide? I could pull that up. Let me
Yes,
see if I can
please. Yes,
reshare this. And this is somewhat complicated, and the mechanisms themselves are, oops. Click here. Let me try this. No, I'm just not. Let me try one more time. I'm just going to go back in and see whether I can pull this up, because it's just not letting me do that. But basically, the actual mechanism that these particular ones, or the combination of how they work together to affect pathways that are involved in ALS are complicated, and these are consistent, but not, Well, it's a good thing it worked before, because it's just not working now. But there are mechanisms and there are pathways, and I think I'm going to leave it at that without showing you the details. This is coming out in a publication that we have. It's under review right now. And our last two publications also talk about how these microRNA are related to neurological processes and to ALS. Basically, we don't know a lot about them because microRNA are so that they can in fact influence many different pathways, and so because of that, they're a little difficult to say this particular one is affecting this process, because it's probably affecting 5 or 6.
Perfect. Next question we have is from Will. Will has lived with lower motor neuron disease for 17 years, and this has now traveled to his diaphragm. The question is, will this test work with slow progression forms of ALS?
So far, we would have to say we think that it would. We have looked, we have some indication. A lot of the samples that come from the CDC don't come with a lot of information on how fast the patients are progressing. They don't have information always on even what the ALS-FRS score is at the time that the blood is drawn. There's sort of, it's a reservoir of samples, but it has some limitations to it. We think so. We think it can actually identify the earliest stages of ALS, and we know that because the initial clinical trial were all blood samples that came at the time of diagnosis. So they were in those early stages, but we think we can actually go back in an earlier stage and identify them. And we have not yet seen any difference between patients who were fast progressors or slow progressors. The microRNA still held true.
Perfect. Next question is, will this work on PLS? I think this was answered already.
Well, we've been working with Dr. Hiroshi Mitsumoto on identifying some unique microRNA that are unique to PLS. So rather than just saying, can we differentiate the two, we want to find out something that's unique for PLS. So if you go in for a test and they think, oh, you have neurological damage, it could be ALS, that we could say it's not ALS and it definitively is PLS. So that's kind of our next step. And he's very excited for us to find these unique markers. We have some things already in process, and we're ready to look at those samples. We only have 42 because PLS is even more rare than ALS. And so getting bigger, larger sample sizes will be critical to this work. But we think we can identify something that is unique to that, as well as just being able to say it is or is not ALS.
Perfect. Next question we have is from Carrie again. Question is, can you use samples from the NEALS biorepository, or can you collaborate with the ALS TDI ARC study for samples?
So yes, NEALS we've been talking to, and we're hoping to get some samples from them. The other one, the TDI, we have not contacted them, and I would guess that they would be willing to, but we need to contact them. I have not done that. If you have a contact and somebody to let me know, shoot me an email or something, and I'd love to have a person to specifically go to, because I don't have that at this point, but they do have a biorepository and we could look into that.
Perfect. Next question we have is from Barbara. The question is, how does the neurologist or other third parties contact you about the blood test?
We are Brain Chemistry Labs. You can find us on our website. They can contact, just go through the website, and an email is typically sent to myself or to Dr. Cox, and we can respond to that directly. So that would be the way to do, is to go through our website, send an email, and then get us in contact. If any neurologist or MD, I talked to an MD today who was worried about an ALS patient or a probable ALS patient, and we're always willing to talk to people. Our goal is to help you. We work for you, and we're willing to do that.
Perfect. That's about wraps up this post questionnaire. Do other people have any more questions before we transition back to McFinn for any final words? Any questions at all, you're welcome to unmute yourself. You're welcome to post in the chat. Please, if you have any questions, please let us know for the speaker before we transition. Oh, perfect. We have a question from on the Zoom screen. It says D-call. The question is there potential for this test to be used in clinical trials as a measure of drug efficiency or ALS progression?
That's a good test, and something that we have plans to look at in the future. We have not looked at it in terms of prognosis, and that's something that we will do. We think it could be used right now as a way to enroll people in the test at the very earliest stages. And that's really important for a drug to work. So we're hoping that could be the first use, is a diagnostic for entry into a clinical trial. But as we parse the data, there may be some use in progression as well. And we were just talking 2 days ago about moving forward with that aspect of it.
Perfect. Any other questions for our speaker? Perfect. I guess if I don't see any more questions in the chat, thank you again, Dr. Sandra Banack, for this phenomenal, wonderful presentation. But we also understand that your time is very valuable, and you have many more wonderful things to do in the world to help us support, encourage more ALS research. Thank you very much. I see McFinn with the hand of applause. We all do a round of applause, and the cheering will follow from McFinn here.
Thank you for letting me be here, and we wish you all the best. And as I said, we work for you. We are a not-for-profit, so we always feel like the patients and their families are really who we work for. So we're here trying to make things better. And we in addition to this ALS diagnostic test that we have, we also are interested in clinical trials and have some data that shows that the drug that we've been looking at can slow progression. We can't stop it, but we can slow it down. And that is really critical, and we're moving forward in that vein as well. And maybe that's the topic of another presentation.
Well, thank you, Dr. Sandra Banack. You're welcome to stick around, but in the meantime, I would like to transition this back to McFinn for final words before we stop recording. But floor is yours, McFinn.
The floor belongs to everyone here. Thank you all. You know, we are powerful because we're together. And Sandra came tonight to tell us that we're powerful because we're together. And we are the people who put the smile on ALS.